So stress is something we all are extremely familiar with. We need no introduction to this. It is something that we encounter almost on an everyday basis. Sometimes it is very trivial, it is minor, like say being stuck in traffic, not having hot water in your shower on a cold winter's day, talking to an audience like this. Sometimes it's more major, it's more intense, it's harder than what you would want, and unfortunately so. For instance, being in a car crash, being in a war-like situation, like how some people are at this point of time, like having to take care of a child with disability, or losing a very loved one in a pandemic. So these are extremely stressful situations, which some of us, unfortunately, have to deal with. But I want you to now imagine a spring. So just like how when you compress a spring, but when you release the pressure, it comes back to its normal state, we have this ability to rebound and come back to our normal state of being after a stressful situation. And this is what we call being resilient to stress. Each one of us have some ability to be resilient to different stressors, but unfortunately, some of us are also very susceptible to some stressors. Being in a state for a long period of time results in conditions like anxiety, depression, post-trauma, and this is more so in women than it is in men. So we started our research asking this question, that yes, we know that each one of us have a different ability to come out of a stressful situation, but when do we actually get this ability? Is it something that develops as we become adults? Is it something that we gain as we are teenagers, or maybe earlier when we are kids, or maybe even earlier when you're in your mother's womb? So to answer this question, we went to the models that are conventionally used in this area of research, which are mice and humans. So scientists use mice in the lab to understand how they respond to stress, or they look at humans and understand how we respond to stressful situations. But in our case, both of these are going to have some issues. And why is that? When you consider young mice or young human babies, they are extremely vulnerable. The other problem is that they are very dependent on their parents in the first few days, weeks, months, or even years of their life. This makes it impossible for us to actually stress these animals or humans and do any experiments with them, right? So we came up with a solution, and this was to use this striped fish, appropriately called the zebrafish. So this is a 3 to 4 centimeter long tropical fish that we use in our lab. The advantage with this fish is that, number one, especially for us, is that the fish lays her eggs and she disappears. The eggs they have to fend for themselves. They hatch and they grow, something like this. I'll try to play this video, and you will see how independently it's developing, very unlike these mammals on your left. So, and now it becomes a baby fish, which we call a larva. The other thing you might want to know in order to appreciate why we chose this model is that this, even though very different in appearance, this fish is about 80% similar to us genetically. So not as different as you think. So what we did was to take these young fish, and I designed this assay where I put one fish in a compartment of a big plate, and in 1 hour, I'm going to record the behavior of about 100 fish in an automated manner and see how they respond to a stressful situation. I'll now show you how these fish respond to a stressful situation. So when you look at the lane on top, those are fish that were not stressed. So they have just come into this compartment. They are very happy. They are trying to look around. Hey, where am I? This is a new place. So you see them in red, which means that they are normal, they are active, they are moving, exploring the arena. What you see below are fish that were stressed just before the recording. And what you will see is that not many of them are really happy over there. They exhibit this behavior that we call freezing. So they just freeze and stay in a place till they feel comfortable to actually explore the compartment they are in. But what you will also see is that these are different time intervals. So each fish, I've shown you how it behaves over time. You'll see that some, like the one in the number 3, is really fast. It behaves like the control unstressed fish as soon as it's put into this compartment. But some others, like say the ones in lane 8 or the one in number 1, take a little more time. So from this, what I want you to understand is that each fish has his or her own way of recovering from a stressful situation. When you look at the first few minutes, so when you look at this is how a graph looks. So when I look at how they are recovering, I see that there is a minority, which is like the number 3 over there, which I'm going to call resilient fish, because they are able to recover from a stressful situation really fast. As opposed to them, a major majority are the ones in yellow, which are going to be called susceptible. What I also saw is that these are features that stay with these fish. So the baby fish keep these features with them as they grow. When they become teenagers, they are still, the resilient are resilient, the susceptibles are susceptibles, and as they grow to adults also. Doesn't stop with that. They also pass it on to their young. So they also make sure the babies get similar properties to their parents. Now, what was bothering us was, okay, now we have the same normal, seemingly normal fish, which have these differences. What is causing these differences? So we tried to look at a lot of things. Eventually, when we started looking at what genes are different between these two groups of fish, we were expecting, okay, the normal way we think is that the brain controls how we behave. What we found threw us off our feet. What we found was this, that there are factors which are involved in the immune system, which are actually different between these two groups of fish. From your school days, you might remember that this is a system which is involved in fighting off pathogen attack, infections. So when you want to come out of an infection, this is the system that's working. Apparently, it does not just that. It also works to decide whether you're going to be resilient to stress or susceptible to stress. To be more specific, what happens is the ones that you saw in blue, the ones that were resilient, the ones that recover really fast, they had this ability to fine-tune the immune system and this fine balance of being able to turn it down and up whenever required. This was what was making them resilient. We proved this using different methodologies, the details of which I won't go into details today. But the message I want you to take today are two. One, our ability to respond to a stressful situation effectively is determined very early, probably when you're really tiny in your mother's womb. And it is influenced not simply because of who you are, it comes from your parents, from your grandparents, and a lot more. The other message that I want you to take home today is that, unlike what you might think, it's not always your brain that regulates how you behave and how you respond to a stressful situation. It's also your immune system. It does much more than simply fighting infectious conditions, diseases. With that, I'd like to thank my lab, my PI, my group who are here, who are extremely supportive, and Noah, with whom I have worked and who is also here today. And thank you all.
Thank you very much for this interesting talk. Questions? Yes?
Yes, when you talk about the resilient versus susceptible fish, do you see differences in the type of stressor, like what you're inducing, and are those resilient fish resilient to those different types of stress?
Yeah, so we used a variety of stressors. So we wanted to make sure that these fish are not resilient just to one stressor. So we tried to use different stressors, and we vary the stress depending also on their age. We are a little restricted when they are young, so we use stressors like taking them out of water, putting them in through osmotic shock. In adults, it's very similar to us. Sometimes it's just discomfort, like making them stay alone, because they're very social, just like us. They don't want to be alone. or putting them in an elevator-like situation, very crowded, very tiny, sometimes more intense. But yes, we put them through a variety of stressors, and they show this repeating attribute or personality trait, as we like to call it.
Yeah.
A great talk. If you manipulate the immune response, can you change resilient into susceptible and vice versa? And if so, how long does the effect last?
How long the effect lasts, I don't have an answer, but this is what we did. We used chemical means of actually perturbing this particular. So to go into specifics, we saw that the complement system is specifically perturbed or is different between these two groups. So we used chemical inhibitors of the pathway. We also used mutants, and we saw that if you perturb these, then it leads to more resilience in a population compared to if you don't. So we checked it with these methods.
Thanks.
Yes.
You said that there is difference in resilience in male and female in humans. Did you see that in the fish too?
Very nice question. So in the young fish that we checked, by that time, gender is not determined in these fish. But as these fish grow, we observed something that was very interesting. In the resilient group, there was a greater tendency for them to develop into males, and in the susceptible, they had an increased tendency to develop into females. So, and if you do an assay on adults, we see that the susceptibles show a greater tendency to be females, unfortunately.
I have another. So when you think about the immune system and you also think about exposure, I'm not sure how it works in zebrafish, but did you try to do any kind of exposure and see if you could get a difference in resilience in the fish for the actual establishment of their immune system when they're young, if you think about it in the context of humans?
I don't understand what you mean by exposure.
So you know the idea of when, as a child or when you're younger, you could potentially be exposed to things that can affect the immune system. Mm-hmm. So I'm curious if there's a way to check that in zebrafish, if there's something that you could do to see if you could alter the amount of resilience.
My answer to that might not be direct because we didn't do the exact same experiments, but the perturbations we used was, like I answered for her question, like genetic and chemical, but not actually challenge them with pathogens and try to see if that affects them. This we didn't do.
So with that, I would like to thank Amrutha.